Hello, and thank you for stopping by to listen to my talk. My name is Sebastian Sanchez, and I'm here to represent the University of Texas at El Paso, presenting my project on the expression and purification of human circadian protein, HROR gamma, for structural and functional studies. HRR gamma refers to human retinoic acid related orphan receptor gamma, or as you will hear me call ROR gamma or RORC for short in this presentation. So, to give a little bit of context on the circadian rhythm is a biological process which controls the sleep-wake cycles of many living organisms. As you can see in this diagram here, the circadian rhythm is a cellular process happening in every single cell of your body, especially in humans. This entire process is driven by the BMAL and clock proteins, which are allowed to heterodimerize within the nucleus. They serve as the positive transcriptional factors to PER and CRY proteins. So when the PER and CRY proteins are expressed continuously throughout the day, they are kept within the cytoplasm where they are allowed to heterodimerize and also accumulate. So eventually it reaches a concentration around nighttime, which allows its translocation back into the nucleus to inhibit the BMAL and clock heterodimerization and thus stopping the PER and CRY production as well, thus completing the negative feedback mechanism. In addition to this cycle that we have here, one of the many accessory pathways to the circadian rhythm include RORs. ROR gamma is especially important because it controls the metabolic genes depending on the certain cell that this circadian rhythm is activated by, as well as support the entire circadian rhythm stability. As you can appreciate through this diagram here, that any disruption that occurs within the circadian rhythm loop or accessory processes can cause a myriad of disorders. This would include insomnia, narcolepsy, mental disorders, even weight gain, and much more very nasty disorders. And so what we aim to do is ameliorate those through many different compounds. Now, one particular compound we're very interested in is nobiletin. Nobiletin is a naturally occurring flavonoid compound, which is found in many skins of vibrant fruits. And what has been shown through previous research is that it's a cofactor to ROR gamma, and it increases its efficiency through a yet unknown mechanism. So we want to find out exactly what happens when we introduce the nobiletin to our ROR gamma. So we've set up this experimental design in order to find the crystal structure relating to this. We first began at the cloning phase, and we've moved to transformation, taking the plasmids that we have made and putting them into our BL21 E. coli cells. We are currently in the expression and purification stage. This is characterized within these two boxes. The first one shows us creating a bunch of liquid cultures and inducing them with IPTG. The cells are then separated from the media and are prepared with many different lysing agents in order to separate the cells from the soluble proteins. We then take those soluble proteins in the purification box and use them in many different columns in order to get our purified samples as fractions. What we plan to do with those samples is run a bunch of different characterization tests, and eventually in the functional analysis phase, we will then add our nobiletin to find out exactly what kind of conformational changes are occurring to our protein. So just to go over our results, we have upscaled the expression protocol very nicely to 4 liters. And in this SDS page here, you could very clearly show that when comparing our 4 uninduced samples with our 4 induced samples, we have grown a very nice and distinct band in our induced samples, which we believe is a successful expression of our RORC. Moving forward from our expression, we have then made many different tests to optimize the solubilization of these proteins in order to survive in the extracellular fluid. And what we have found here in this STS page is that freeze-thaw methods are a lot more efficient at solubilizing our protein over sonication. But once we have prepared our sample and we are allowed to use those soluble proteins and run it through a His trap column in affinity chromatography. We have three main washes and one main elution. And as you could tell in this in this SDS page on the right, that our main elution happens at 180 millimolar step gradient of imidazole. What we could see here is that at this solution, we have a very nice and thick band, which has shown very efficient purification of our RRC, but it still needs a little bit more purification to go through. So we end up taking those samples from the main elution and run it through a size exclusion chromatography. In this chromatogram here within the highlighted region, we end up running these samples on our SDS page. And as you could see, we have a lot of very concentrated, very pure samples of proteins that we have collected as a result of this size exclusion chromatography. So delving a little bit into the characterization step, we end up taking those samples that we have collected and perform some circular dichroism. What we have found in the CD data is we have a very distinct trough formation, and this trough is very characteristic of alpha helices. This is very good news for us because as you can see in this crystal diagram of ROR gamma, it is mainly consisting of alpha helices. So what this demonstrates to us is that its secondary structure for our sample has been maintained throughout this entire experimental process. So just to go over exactly what we have learned in this project so far is that ROR gamma, the expression plasmids were successfully transformed into our E. coli BL21 cells. The expression protocol was then optimized so we can obtain a lot of these proteins. We have effectively solubilized our protein using the freeze-thaw methods. And finally, our protein was purified using affinity and size exclusion chromatography. We have then taken those samples and run a bunch of CD tests, and we can confirm that the sample has maintained its secondary structure, which are the alpha helices. What we have encountered as a result of many of these purifications is that the large-scale purification works best in obtaining these very high-quality purified samples, but it poses a challenge to our instruments because of the presence of contaminations, which we believe are lipids. So one thing that we can try to do to further these experiments is to minimize the lysis protocol in order to minimize these contaminations that we are finding. Speaking of future directions, what we plan to do with this project further down the line is to continue expressing and purifying our HROR gamma in order to accumulate much more protein to work with. We then want to further optimize the affinity purification techniques by saturating the column and refining the solution gradient, and also find out the more CD data of ROR. What we plan to do is study the CD data at different temperatures in order to build and characterize this thermal stability of our protein. Furthermore, what we plan to do is introduce digestion enzymes in order to remove the His tags from our recombinant protein in order to prepare it for further purification techniques such as ion exchange chromatography. And finally, when we obtain our officially purified sample, we want to introduce our nobilitin in order to find out the conformational changes which we expect to occur as a result. This concludes my presentation on the circadian rhythm protein HROR gamma, and I hope to hear your questions in the near future. Thank you for listening.